Welcome to the St. Joseph College open web-based platform that is Sao A Learn. And also welcome to the Department of Biotechnology, St. Joseph College for Women, Autonomous Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level. The course Plant and Animal Biotechnology comprises 5 modules. The first module is plant tissue culture techniques and secondary metabolites production. Within the first module, the chapter 4, that is application of tissue culture, under this heading, micropropagation will be discussed in this chapter. In this video, will be discussed by Dr. A. Veerabrahmam, HOD and Assistant Professor, Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam, Andhra Pradesh, India. The chapter Micropropagation has the learning outcomes are as follows. The students will be able to learn, will be acquainted with the concept of highly in vitro process. Since the micropropagation is completely, it's an in vitro process. Okay, uh, it's strictly applicable in within laboratory process, and highly aseptic at high aseptic conditions only the micropropagation process will be obtained, and the students will be able to evaluate the important stages in micropropagation, so which comprises three different and important stages, that is establishment, multiplication, proliferation, etc. And we'll be able to determine the factors affecting micropropagation, able to design multiplication and proliferation cycles with economic crops. Since its commercial application of the micropropagation, so many economically important crops can be regenerated without any pest and without any effect. And also the students can able to reproduce the protocols for pilot scale operations by adopting the technology, that is micropropagation. So students can reproduce the protocols for pilot scale operations. So these are the contents for micropropagation chapter, that is introduction to micropropagation, stages of micropropagation, media preparation and sterilization, initiation and establishment, multiplication and proliferation, one of the very important key aspect of the micropropagation, and lastly application and commercialization of the micropropagation. So let us see what is micropropagation. It's a type of clonal propagation. It's a process of creating new plants from small plant parts, that is micro. The name indicates the micro means a small, of course, maybe I mean sexual parts or floral parts or apical tips, root tips, apical meristem, lateral buds, apical buds, leaf primordia, any of this. Because of the Haberland concept, that is a totipotency, the micropropagation ensures the establishment of a variety of plant species of our choice in a sterile environment using tissue culture techniques. It is an efficient way to produce a large number of identical plants in rapid condition, ensuring genetic consistency. So every time the process look forward for to produce large number of identical clones, identical plants. Of course, there is no involvement of the sexual reproduction. The identical clones can be produced. Generally, in case of plants, higher plants, due to the sexual reproduction only, the identical clones or different clones will be obtained. Of course, in those case, even though it is a sexual reproduction, no, the clones may not be produced in a rapid condition, but here in micropropagation, the clones can be produced in a rapid condition. Of course, if the cultural conditions and factors affecting the micropropagation followed, observed at close proximity, then only the products will be prominent and also promises the better life in the society. Micropropagation that offers benefits, wonderful benefits like disease-free plants. Disease-free plants, since they are raised inside the aseptic conditions and also in vitro conditions. As the name indicates, the in vitro means aseptic conditions, maintenance of the um, uh, asepsis and high genetic consistent clones, identical clones on a defined medium, etc. So the ability to propagate rare or difficult to root species can be propagated through the micropropagation. So let us see what are the stages. So of course, before going to the stages involved in the micropropagation, what is the exact meaning of the micropropagation in Greek? So micro means a Greek word meaning a clone, clonal, and a twig. Identical copies can be obtained in aseptic conditions. So multiplication of genetically identical copies by asexual, not a sexual reproduction, that is called clonal propagation. Of course, the other name for the micropropagation is the clonal propagation, and it is completely asexual reproduction, and multiplication occurs on individual or a group of or a genetically identical copies, and multiplication of genetically identically individual plant species or individual cells or aggregates by asexual reproduction, right? And so very little bit of time, a short period of time, the plant species can be propagated in indefinite manner. So let us see what are the stages involved in the micropropagation. So first of all, it involves the four important stages, that is in vitro establishment, multiplication, rooting, acclimatization. So each and every stage is very important and significant in consistently maintaining the micropropagation in vitro condition. So in vitro establishment, let us see, the explant is placed in sterile culture media and begins to grow. So the propagation starts with the inoculation of the explant, which is excised from the mother plant. Any excised portion or a part, tissue, or a cell, even aggregate or a tip. So that could be an explant and can be inoculated onto the culture media. Maybe so most probably it is a agarized solid medium. So prior the establishing the plant part, the plant must be surface sterilized with a variety of chemical sterilants and also followed by distilled water sterilization, and prior the sterilization, media must be prepared involving the major and minor nutrients, and carbon source, that is the sucrose, will be added and agar is added, and then it is sterilized on in autoclave at 121 degrees centigrade, 15 to 20 minutes duration, 15 lb pressure will be applied. Of course, we know very well about the main principle behind the autoclave. So that is the boiling point of the water generates the water vapor that hits the surfaces of the culture material, and as well as the culture media also be sterilized. Of course, in previous chapters we discussed in detail about the autoclaving, culture materials and media and all. The next phase in the micropropagation is multiplication, in which the plantlets are repeatedly subcultured to induce growth and multiplication. So here the multiplication of the plantlets, especially here, a short span or short period of the callousing, because callus may not be subcultured number of times. So only one or two times it has been subcultured, and the subcultured callus aggregates will be induced under the um, um, hormone ratio, that is cytokinin rich and auxin less concentration. So the plantlets are regenerated by subculturing the small aggregates of the callus, and so the growth and multiplication of the uh, lateral buds or multiple shoots will be erased on multiplication stage. In the next stage, that is rooting. So this is completely after um, evaluating the complete multiple shoots or adventitious shoots because of the cytokinin to auxin rich uh, ratio enhances the multiplication of the multiple shoots from the lateral stages or apical stages of the cell aggregates or callus aggregates. So then induced to enhance the rooting at the base, that is at the radical stage. The plantlets develop roots in a separate rooting medium. So in rooting medium again the cytokinin concentration will be reduced, and auxin concentration will be enhanced, especially IBA concentration has been increased, indole butyric, so that is the hormone which can enhance the rooting process. And then the fourth stage, that is acclimatization. So after all the plantlet grown at a stage of the root stem, that is adventitious shoot and a little bit of propagules, a leaf primordia obtained, then the plantlet should be acclimatized to the normal conditions. So that in this regard the rooted plantlets are carefully aseptically cut down. Of course, after rooting, every multiple shoot is cut down and again inoculated onto the MS medium, supplemented with cytokinin rich and auxin less concentration to enhance the number of clones. So in this process, every time uh, the clones can be increased. That's why it is called as basically it is a clonal propagation. Okay, so rapid multiplication, multiplying the stock plant material to produce a large number of progeny plants using modern plant tissue culture methods. Okay, and after the rooted plantlets are gradually introduced to the greenhouse chamber or greenhouse environment, so it will be termed as a acclimatized to the conditions. Of course, it is not so easy to, as we discussed, the acclimatization of the rooted plantlets, since the each and every plant species requires a variety of special conditions and factors that influence all these four stages in the micropropagation. So it involves several distinct stages. Each stage contributes to the successful production of new plants, and it is a delicate process sometimes in case of banana clones or other floral species requiring careful attention to detail. Otherwise, degeneracy or leaf curling, multiple shoots may not be originated, right? Yeah, now let us see what is the media and how the media will be sterilized. All the media contains nutrients, vitamins and hormones essential for plant growth and development are incorporated or suspended into the distilled water, and the composition varies depending on the plant species. Every time based on the plant species you have to design the components, or especially vitamins and hormones, right? And then preparation. The media is usually prepared in a clean and sterile environment to minimize the contamination. Yeah, in culture room where the already room sterilization is carried out with the help of fumigation. Formaldehyde and potassium permanganate crystals are used to rise the invisible fumes that clears the any air distributed fungal spores are there, will be controlled, will be cleared, so that in culture room it is free from contamination, so that you can carry on the preparation of the media preparation. And this typically involves dissolving the ingredients in distilled water. The distilled water is most probably all the commercial agencies or frames uses the millipore cube water, where the which is filtered through 0.02 micrometer in diameter sized filter, and adjusting the pH to 5.5 to 5.8 is the optimal range of the pH for the media preparation. Then the next step is the sterilization. Of course, we discussed more detail about the sterilization, that is either is a moist heat or a dry heat or a filter sterilization. So here, since it is a media, liquid media, which uses a high pressure and temperature that will be attained through the autoclave or in a pressure cooker to eliminate any microorganisms that would be attached onto the surfaces. And of course, the plant can also be surface sterilized with the help of chemical sterilants like sodium hypochlorite, magnesium hypochlorite, calcium hypochlorite, and bevastin also can be used, whether the carrot cambial tissue used as a culture material, then all the carrot cambium tissues must be washed with the bevastin as a fungicide. If any indigenous fungal spores are involved in the tissue, so they may be completely removed from the stellar tissue of the carrot, so that after one round of the bevastin, maybe 0.01 to 0.5 microgram concentration per ml will be used for surface sterilization, and then followed by distilled water sterilization two or three times in order to control these traces. Any traces will be there, so these traces must not be there on the tissue, especially on the stellar tissue of the carrot cambium if the culture material is carrot. So let us see how the initiation takes place and establishment. The stages marks the beginning of the micropropagation process. So sterilization. So explants are sterilized. Already we discussed just now the prevent contamination. Culture media. Sterilized explants are placed on the culture media. So usually MS medium, Rashigi and Skog medium, supplemented with the BAP, benzyl aminopurine, or the natural cytokinins. Best example, kinetins can be used for establishing the micropropagation. And growth hormones that promotes the shoot and root development, especially first of all a little bit of callus intervening, and then the multiple shoots are raised because of the increased concentration of cytokinin, that is BAP, benzyl aminopurine, and of course kinetin, zeatin can be used. And then callus formation. The explants are allowed to grow and form a little bit of callus. That is the initial phase of the micropropagation. So establishment occurs when the callus forms shoots. So when can the shoots will be formed? When the auxin concentration, of course here you have to formulate or evaluate whether the explant containing any indigenous auxin concentration is there. You need to evaluate. So by a variety of chemical reagent tests, we can evaluate the concentration of the indigenous concentration of auxins. So since they are the tryptophan derivatives, we can easily test with a protein quantifying reagents can be used to identify whether the auxin rich or not, right? Yeah. So callus can be established from and then callus is induced to form shoots, roots, and eventually a plant. The next stage, that is a very important stage in micropropagation, that is multiplication and proliferation. So multiplication is very important in micropropagation on a commercial basis, since after all the callus is induced to gives rise to a lateral shoots. Of course they may not be matured or having the at least each multiple shoots having a 2 to 3 lateral buds than the apical buds. So that is the basis for the establishing the multiplication of the a particular or a desired clones onto the defined medium. So that every time the technician or the students undergo the subculturing of the callus aggregates to obtain the multiple clones involving transferring the growing plantlets to a fresh nutrient medium. So here choosing the fresh nutrient medium is very important, and it's very significant for further development, and the subculture is done periodically to ensure optimal growth conditions. Then shoot multiplication. So through the use of plant hormones like cytokinins, especially benzyl aminopurin and kinetin, either of the can be used as for the shoot multiplication. The process encourages the formation of multiple shoots from the initial explant. So this step is crucial for mass production or mass multiplication of the desired plant species or plant material on the defined media. The next phase is proliferation. This phase involves the repeated subculture and shoot multiplication process, leading to the generation of large number of plantlets. So that will be ready for the next phase of development, that is acclimatization to the ordinary conditions. Yes. So after successful multiplication, the micropropagated plantlets are ready for rooting. So here again the hormonal concentration must be designed, or have to observe the what hormones are required for the rooting purpose. Generally for rooting, an auxin can be used in much concentration, maybe IBA or IAA or naphthalinic acid, NAA, can be used to enhance the roots onto the shooted plantlets. So process involving transferring the plantlets to a rooting medium where they develops roots. Different method exists for rooting, that is including the use of auxins, increased concentration of auxins, say maybe natural auxins or synthetic auxins, example 2,4-D, 2,4-dichlorophenoxyacetic acid also used, and specialized rooting media. So that is incorporated with the IBA, indole butyric acid. So the further proliferation involves the acclimatization, that is a gradual transition to outdoor conditions. So once they are acclimatized to the greenhouse chamber cultural conditions, so it is the right moment to transfer them to the outdoor conditions, since they must be acclimatized to the ordinary or field conditions. And then next step in the proliferation, hardening stage. So increased and frequent exposure of the plantlets to the sunlight and wind to withstand the ordinary conditions. Otherwise prolonged maintenance of those plantlets even after they acclimatize to the greenhouse chamber, it is very expensive process if still you are maintaining in the greenhouse chamber. So it is very expensive process. So that is another factor. The next, further rooting will be induced on a suitable only rooting medium, so that is a suspension medium. So once the plantlets are rooted, the plantlets need to acclimatize to the outdoor environment. So that involves the gradual transition from the controlled conditions like any mist chamber or a greenhouse chamber or polyethylene chambers, uh, and lab to the harsh conditions of the field. Hardening often involves increasing exposure to the sunlight and wind, preparing the plants for survival outside the that is, specialized equipment is required, facilities are required, and more technical expertise is required, because every time close monitoring is required by the experts, by the technician, or the agriculturist or tissue culturist. Protocols not optimized for all species. This is of course one of the biggest disadvantages with the micropropagation. Only some plant species the micropropagation has been um, optimized, the protocols were optimized, and plants produced may not fit for industry standards. Some plant species, but not all these plant species, and but relatively expensive to set up. Of course, the initial setup is somewhat expensive in order to establish aseptic conditions, aseptic operations, instrumentation, and ensuring the ISO guidelines and GLP guidelines if it is a commercialized aspect. So these are some disadvantages with the um, clonal propagation or micropropagation.
Let us summarize the present chapter, that is micropropagation. So one of the best application of the plant tissue culture. Micropropagation is a method of rapidly multiplying plant material to produce large number of progeny plants using modern plant tissue culture methods. And this technique is widely used for propagation of various plants, including those plants can give rise to a variety of alkaloids, flavonoids, terpenoids, gums, resins, and saponins, and other essential oils may be used as secondary metabolites and very benefitable to the human society, and also a commercial purpose, can be extensively grown or obtained through the micropropagation. This technique is especially applied or integrated to those that are difficult to propagate through traditional methods. And the next one, the key steps in micropropagation comprises selection of explant. So this is a important criteria where the explant condition, probability, health, and hygienity is very important to establish the micropropagation. And then sterilization after selecting the explant, after completing finishing the media preparation, either way of the autoclaving or dry heat sterilization, all the materials will be sterilized. And then next followed by initiation of the culture, where the plant material as an explant is taken from the mother plant, will be introduced onto the defined medium. Initially an unorganized group of cells will be raised, that is known as callus, and then followed by multiplication, where the hormonal concentration, that is auxin to cytokinin ratio, will be varies. So here the cytokinin ratio has been increased to get the multiple shoots, that is multiplication, and then rotting process. Here the hormonal combination, that is auxin to cytokinin ratio, will be reversed. The less concentration of cytokinins and high concentration of auxins will be used for obtaining the routing, probable routing, and acclimatize into the laboratory conditions. And finally, acclimatization. So this phase is somewhat difficult to acclimatize to the normal conditions, where the rooted plantation, rooted saplings or seedlings are transferred to the ordinary conditions, that is transport to the greenhouse chamber or polyhouse chamber to acclimatize to the normal conditions. Overall, micropropagation is a powerful tool in modern agriculture, horticulture, and conservation efforts, offering numerous benefits but also presenting certain challenges that need to be managed effectively. And these are the references for the present chapter, that's micropropagation. Go through each and every reference to get more knowledge and more clarification about the present chapter. Thank you for great attention in the present chapter.